Thanks very much. Thanks for those generous remarks. It's been a pleasure working with all of you over these years. I think we want to move right along because these are what the agenda that Drew's laid out is a really important one. And I think we'll find that we're going to run out of time. I hope you find this as compelling a session as the past 2 days have been. So we're going to initially just march through the list of talks here, beginning with Steve Moore's presentation, and then move into an open discussion framework when I assemble the panel up on the stage. I don't have biographical information on each of our speakers, so I'm going to ask that each of you begin with some introductory remarks about who you are, why you're here, why you're interested in this topic. So let's kick it off with Steve. Thanks.
That was the hard part. Okay, so I do the Ross Perot Vice President thing, explain who I am. I'm with the Goldman School of Public Policy. And we had a very pleasurable task to try and map what interventions the community could meaningfully do at this level, both in terms of things that people are generally predisposed to do and things that would have then an impact in the world. And so this talk is to sort of lay out that landscape. We're not going to cover any large fraction of that landscape today, but I hope that some of you will walk away thinking, gosh, that's kind of an interesting idea. Maurer got it wrong, of course, but I can improve it, right? Because the thing about all things political, including looking for consensus in a community like this, is originality is bad, right? This is a thing that academics resist. You want to do something that's sort of sensible that everybody knows, and if you can actually do it, that's already an achievement. So we were trying to see what we could do to facilitate governance in this field, and I just want to give you the quick version. The long version for people who want it is out in the hall. There are hard copies of the report if people like to leaf through, and I'd love to hear from you. So the purpose of the day, so I'm told, is governance, and that's a little bit like Mark Twain's observation about the weather. Everybody talks about it, nobody does anything. And I want to congratulate you for coming to the governance session, and hopefully there will actually be a little governance from this, which would be a wonderful experience. But if you are thinking about doing governance as opposed to admiring the word, what does it mean? And it seems to me it means these two things anyway that you can unpack. The first one being that people have studied this community, have studied the biosecurity issue for a long time now, both in funded and in unfunded ways. You have about 5 years of talking about this stuff. And there's sort of two reasonable things you can take from that process. The first one is that you surely know some things at this point, right? And the other one is that you surely haven't solved the entire problem. And the question is, can you take the things that you now know securely from this large investment in thinking about the problem and actually turn them into actions that make things better? And I'm in the public policy business. I don't believe there are any silver bullets. You don't fix things absolutely. If you make them better, you congratulate yourself and go home. You do a nice day's work. So there's more mechanics in the middle of this. You need to turn things that people reliably know into something that everybody has demonstrated themselves is true, a consensus. You need to turn that into a declaration, and then you hope that the declaration actually leads to meaningful action in the world. So what did our project do? We wanted to find out what the knowledge was out there and to help build some consensus. And what we did was we interviewed 25 people in the community, about an hour each, and thank you, several of you are in the room. We then wrote a white paper to say what we thought the lessons were of what people had told us, and then we circulated it and got feedback. The white paper, by the way, also has some very nice attachments, short white papers by Professor Zoloth and others. Ray Zelinskas talking about sort of specialized topics that you'd really want to have your hands on to think about biosecurity issues, things that the average synthetic biologist or the average public policy person doesn't know. Then we held 2 town halls and discussed these things, and you can actually download them on videotape. I think it's a really interesting social exercise to watch people try to say what's the space of things that we know are good and can agree on. And then finally, we're going to have this panel discussion, And I don't believe actually that biosecurity is a hard subject, but it crosses disciplines. And so it's hard for one community like synthetic biology to kind of know that they're doing the right thing. And I think one of the reasons to have a panel up here is that these people are on tap. So I hope the discussion is mostly about you guys talking to each other, but occasionally we may be able to tell you something that you don't know because, look, this is about weapons among other things. There's a long history of that. Being a synthetic biologist doesn't teach you a lot about how to think about weapons issues. So what I did in the project then is I identified, I and my co-author, 6 items that are plausible candidates in the sense of having wide interest in the community, wide support, and also seem to be things that would be effective in the real world. And as I say, if you want, you can read the report, but I just want to go through them quickly. So the first one is, the first 2 actually relate to the screening problem, which Drew mentioned already. And the question is, what kind of intervention could you do to make screening better? Well, right now, most screening companies do some screening. They have a full-time employee who basically looks for orders that have something nasty in them. And this employee, is doing a good thing. He may be doing it in a clumsy way, we'll come to that, but they're spending money to make security better. Unfortunately, this is just most companies, right? And there are a few who cheat. And this is actually a familiar problem because you could put pressure on the ones who cheat to join the cartel, if you will. It's an ugly word, but to join the club of people who actually do screening. And if they did that, then you would be going to 100% of the company's screening, and the economics of that is really nice because nobody loses. If everybody does it, nobody's punished. And could the community pressure the few remaining holdout companies to say, you should start screening too? Well, I think one obvious intervention is you could pledge not to do business with companies that don't screen. And if you think about it, the cheaters are only picking up the value of one full-time employee. The amount of business from the community is already comparable to that. You can reasonably exert that pressure. But even if that wasn't enough, presumably the biotech and pharma companies would join in if you said, we really shouldn't do business with companies that don't say that they screen. That would be reasonable economic pressure. And economics, unlike law, is international. So you don't have to worry about the fact that, gee, only blue herons in the US. Economic forces tend to get everybody screening everywhere, and that's a nice intervention you could do with a declaration. The second thing you could do is you could create a better list for these people. They're already spending the money to screen. Let's give them powerful lists. One day, I'm confident the government will get around to telling people, here's the correct screening technology you should use, go use it. But that screening technology will come from people in this room. So there's no particular reason why you can't get on with it and just try and develop that screening technology sooner. And if somebody, by the way, does that, you can say, this is a good screening technology. The companies will be more likely to adopt it if you give them that cover, right? So that's something you could do. The second thing you could do is you could have a place where if I'm about to do an experiment of concern, I can consult somebody. There have been several NAS panels that say there needs to be a person you can pick up the phone and talk to if I'm about to do something I think might be an experiment of concern. Again, this is something that eventually NIH is working on. They're going to put something like this in place eventually. But look, the advice could be available today because even when NIH does this thing, who are they going to call? People in this community. You could start doing it. And by the way, that will teach you something about experiments of concern. There are limits to what you can learn by just sort of having 5 smart guys in a room. If you really want to know what experiments of concern are out there, hang out a shingle and say, we'd love to give you advice about the thing you're thinking about doing. That's how you learn about problems in the world. You could bring more data into the problem and get past it's the next level in researching and understanding what the concerns really are. The next thing you could do is, people are forever kind of waking up in the middle of the night having a new worry about what might be possible with synthetic biology, and they notice things occasionally in the lab like, gee, that's an accident I never thought would happen. And you would want to collect those things. The Federal Aviation Administration does that for airline safety kinds of issues. The system works really well. And when they notice that the same issue comes up 3 times, they send around an email so that other people don't discover it by accident. It's a sensible kind of thing. It's something that could easily be operated in the community. If you guys were to endorse that activity, there would probably be people willing to set one up. I'm not going to touch the next one, not because I don't think it's interesting, but it's beyond my capabilities and time limitations. There there is this question about if you know or think you know that somebody's doing something disturbing, should you tell somebody or should you look into it? And Professor Zoloth will talk about that. She also, as I say, has this paper in the report where you can get her thoughts at some length, so I commend that paper to you. And finally, you know, physics bodies always talk about the top 10 experiments for the next few years. I suspect that if you were to give your collective judgment that there really are some safety and security technologies that ought to be developed in synthetic biology, the funding agencies would listen to that in a big way. So it seems like when you talk to people, there is consensus around certain technologies. If you really do believe that you know how NIH should spend its money, that something's a priority, saying something in this fashion would be a good thing. All right, so where is all this going? Well, you know, the smart answer is to tell you, well, I'll tell you at the end of the day how governance is doing, but I think this is a wonderful experiment, and I'm glad that we've gotten this point. And the longer term issue is I think it's really important that you do things in this mode, because not for sort of the tactical reasons people tell each other stories about, well, if we do governance, maybe NIH will do this or will do that or will do the other. It's because it's the right thing to do. We're all responsible as individual human beings. We're responsible as a community. If we know that there's something that ought to be done, the simplest way to express that morally, Lori can check me on this, but the simplest way is just to do it. And I can guarantee you one thing, if you can bring some step in governance mode, if you can do something in governance mode in this democratic fashion, it will be the least painful way to accomplish your goal, right? There's something intrinsically elegant about asking for something that everybody has to agree to have it happen, to have consensus before something happens. If you can fix problems in that mode, you should try to do it. So I think governance is a great idea. Eventually, I think you'll want to do governance in IP and everything else, but this is a nice experiment. I congratulate you on having the willingness to try it, and I look forward to watching the experiment and anything I can do to help. So these are, you know, what we identified as sort of the 6 things that you might want to think as self-governance in biosecurity. Tweak it, change it, set up an experiment, but I hope, you know, we've made a start on it, and I give it to you now. Thanks so much.
That's great, Steve. Thanks very much. I forgot to add that we're just going to zoom through all of our morning talks first, and then we'll take questions at the end. So Rob Jones is up next from CRAIC Computing. And again, you got to tell us the provenance of CRAIC, right? That's going to be an important opening remark.
You bet.
Okay.
Thanks. The company, which consists of just myself, is called CRAIC Computing. Crack is Northern Irish slang for having a good time. My background is as a molecular biologist that's transitioned into bioinformatics and computing. I've worked in academia. I've worked in the high-performance computing, or for a high-performance computing company and for a biotechnology company in Seattle. And since the end of '97, I've been working as a consultant for a number of different biotech companies, helping them with data management and data analysis. My reason for getting interested in sequence screening was both just a general interest in the topic, but also my colleagues at Blue Heron. The people at Blue Heron are people that I worked with at Darwin Molecular, a company that we set up in the mid-90s. So I'm going to give you an overview of the technical and operational challenges that are involved in sequence screening. Just as a sort of initial background to that, the DNA synthesis industry, both at the level of oligos and gene synthesis, is really a unique and fascinating industry. Ordering oligos, you can go to a website, type in your sequence, give a credit card number, and that triggers a remarkable piece of really advanced synthetic chemistry, and you get the result of that back by FedEx 24 hours, 48 hours later. The convenience and the economics of that has really driven, I would argue, the vast majority of DNA synthesis through a relatively few companies and university core labs. So there's this great concentration of data flowing through a few locations. That poses a risk for those companies in terms of at the level of gene synthesis, potentially biological safety issues, but certainly legal and ethical concerns regarding the materials that are coming through their lab. So the idea is to screen those sequences for matches against a database of pathogens. Pretty simple idea, but it really faces some technical and in particular operational challenges. I'm not going to talk much about the software. It's basically a bunch of Perl scripts that wrap around the BLAST sequence comparison software. Requests coming into the system are coming through various ways, and BLAST is run against a database of select agents. The results are processed, some criteria are applied, and if something nasty pops up, then an email alert is sent out. My screenshot didn't come up, doesn't matter. On the technical issues, the first one is the issue of what sequences do you screen. You don't want to screen against all of GenBank, both for performance issues and the concern of your customers that you would be disclosing or finding out information about the sequences that are being submitted to you. We're only going to screen against pathogens. And there are two approaches at either end of the spectrum. We could build a very small, curated database of genes that we know are of particular concern. Smallpox, anthrax toxin, Ebola. If we did that, we could be pretty sure that anything that matched to those with a high degree, with a high score, would be something that would be of concern. But it takes expertise and effort to create that database. And it also makes a big assumption about the potential threat. If you're only going to screen these things, if someone's working on something that you've not considered, you're never going to see it. An alternate approach is to just take all sequences from all select agents. And that's what I'm doing at the moment. And part of the reason is a practical, is kind of practical advantage that I can write a little script that trawls through GenBank, looks at the organism tags, and just automatically pulls out all those sequences. In doing so, I can, in principle, detect any work involving select agents, whether it's involving a particular anthrax toxin or whether it's involving some other part of the bacillus genome. So in principle, it would detect vaccine work. But the problem is, because I'm really casting such a wide net, I get all of the housekeeping genes, and in particular in bacteria, everything that's highly conserved. So high false positive rate. I think the ideal solution is going to be screening against all of those, but weighting some parts of those genomes and some components therein as much more significant than others. Yeah. And here it is. What I've done, I've pulled everything from the CDC select agent list, the USDA list, and in particular the Department of Commerce Export Administration regulations. And if you look at the numbers of species on here, you'll notice that by far the large number of species are in the commerce regulations. Depending on where your order has come from, so if you've received an order from a lab in the US, you don't care about commerce. If you've received it from overseas, then you do care about commerce. Depending on what you find and where the order comes from, it can influence the action that you might want to take. One way to reduce false positives is simply to use a very high cutoff score. I think in some cases that's not sufficient. If the black line is your pathogen sequence, if these are sequences that have been submitted, if the match is across the entire length, then that's obvious, that's a good match. If it's across a segment, then that may well be a good match. But if it's something else flanked by two small regions, let's say you're trying to insert a gene into a genome, maybe a high cutoff score wouldn't get that. And then if it's a gene where, say, the third position of each codon has been modified, you might not see that. One way to deal with matches from conserved or housekeeping genes, which is a particular problem for sequences submitted that have come from other bacteria, is to screen everything against the pathogens. Anything that comes up positive, screen against a reference database of non-pathogenic bacteria. And so, for instance, a ribosomal protein from one bacteria may well match anthrax and plague, but there may well be a higher score to a non-pathogen. And so that sub-differential comparison may resolve some of those. Another way is to pre-screen each of those pathogen sequences, and you might run, say, a sliding window across each sequence, compare that sequence word against all of GenBank. And if it's a unique sequence, you give it a high weight. If it occurs elsewhere, you give it a lower weight. And that way, when you match a customer sequence against that, you can weight the confidence in that match. Doing that avoids having to screen your customer sequence against all of GenBank. The real problem comes from oligos. Shorter sequences, higher false positives, and as I'll mention in a moment, some real operational issues. And manual assessment of those results may not and probably is not feasible. And so we may have to change the approach there to, rather than alerting when we find something of concern, really just log lots of these low confidence matches. And an analogy there is like closed circuit TV. And apparently these days, if you walk around London, your face appears on 50 or to 100 CCTV cameras. My impression is that that's not very good at preventing crime, but it's very, very effective at investigating crime. And there have been some very high-profile cases there. And arguably, there's a deterrent effect. So screening at oligo companies may not alert us to problems, but it may be very useful for picking up or for investigating after the fact. The operational challenges, and these are kind of the thorny ones. The customer-vendor relationship really is unique in the gene synthesis and DNA, well, particularly oligosynthesis relationship. And there's an assumption on the part of the customer that not only will your sequence information be kept confidential, but you as an oligo vendor will not attempt to figure out what that sequence does. And I think it's a dirty little secret of intellectual property management in biotech companies that often at a very early stage in the invention of something, those sequences are sent out of the company to oligo houses. Anything we do to change that relationship has got to be handled extremely carefully. There's a real risk, if we're too heavy-handed there, of customers saying, well, I'm not going to send anything out, I'm going to do it in-house. And then immediately the benefits of screening have disappeared. companies are already up and running doing this stuff, and anything that we do to deploy this software has to be able to integrate with existing databases and fit into existing high throughput lines. But the big problem for companies is that, given that there is a false positive rate at some level, how do they assess those matches to really tell if it's a true positive or not? And to do that, it really takes some expert assessment, even in gene synthesis companies with relatively low throughput compared to oligo companies. It's a hassle, it's a judgment call, and it's an expense. We've heard over the weekend about the low cost of doing gene synthesis and DNA synthesis, and that's great for the research community. The downside is that that really is a very competitive industry. The margins, the profit margins that these folks are making are really very, very low. Any additional expense, unless, as Stephen mentioned, unless that expense is spread across all players, can really put you at a competitive disadvantage. And the big concern that, the question that all the synthesis vendors ask when you talk to them is, okay, we find something, what do we do? And at the moment, that really is an open question. The first thing you have to ask is where that request came from. So if you're a company in the US, it came from outside the US, then you're looking at potential interactions with Department of Commerce or the CIA. If it's from within the US, you don't talk to those folks, you talk to the FBI. And as I think it's clear from anyone that's had to do any of those interactions, it's really difficult to figure out who to talk to. The next question is, who is the customer that's made the request? And I split these into three categories, the good, the bad, and the foolish. The good is a legitimate researcher who's working on vaccine development, maybe registered with the CDC. The challenge there is for the synthesis vendor to know that that person is indeed legitimate. I mean, they may say, well, I'm registered with the CDC, but as far as I know, those CDC lists are not public. There needs to be some way to verify that a customer is certified and certified and is a legitimate customer. The bad is obviously someone doing something nefarious, and there you definitely want to, if you believe the customer to fall in that category, you want to have a way that you can contact a regulatory agency. The foolish is hopefully a small category, and that may be someone who either out of ignorance or oversight has thought it to be a good idea to use a toxin from one of these agents coupled to an antibody as a therapeutic, and doesn't realize that they need to register with these agencies. And that's the sort of person where you really want to, if you see something, you want to pick up the phone and just have a chat with them. But you don't want to do that with the bad guy. Now, there's also the issue of okay, you found something and you want to report it to one of the agencies. What are you actually doing? Are you reporting a crime? Now, export regulations are pretty clear as regards full genes. The CDC, USDA regulations are clear about full genes. So I think that part of it is, if what you found really contravenes one's regulations, that's a clear thing that you're reporting. But those regulations can be ambiguous, and I think there's concern here that they're definitely incomplete, in terms of how they relate to genes. And then if you're not reporting a crime, what exactly are you reporting? well, to some degree, what you're doing is reporting intelligence to FBI or whoever. And I think that is something that we're not used to doing. I think it's something that, from a corporate legal perspective, kind of raises a bunch of red flags, and it's something that we need to think about the implications of that. And all the recent concern about phone companies turning over records to the NSA, I think that's a good example of those concerns. All this ambiguity really raises concerns amongst the legal folks about liability. And if you don't do any screening, if you don't do any screening and you make something that then ends up in something nasty, then there's potential liability there. If you do screening, in particular if you find something and you're not able to find the right person at the FBI to report it to, or if you sit on that information, then there's also a big liability there. You're damned if you do, you're damned if you don't. And I think that's an open issue. The idea of a hotline, a single place that you can go to talk about these issues, great idea in principle, but I don't think the agencies are anywhere near ready for that. And I think it's critical to engage them in this debate, and the Sloan study is definitely doing that. The NSABB, I think, is a much more formal structure for doing that. I'm concerned that the people that come to those meetings, and I've met a few of them, they're smart, they know what we're talking about, they're quite sympathetic, but they're not the people that are going to make regulations. And they're not the people that are going to take actions. And we need to be talking with the people 1 or 2 levels above them. And until we do that, then there's a risk of, I think, specific risk of if we push for a hotline, then we're going to get a government response, and it's going to be exactly what we don't want. It's going to be very harsh kind of regulation. So to conclude, there's still some open issues, technical issues on screening, and clearly there's operational issues. I think the glass is half full, and I think we have to start considering a different model for oligos. There are a lot of scenarios that I can come up with, that people here can come up with, of ways that you can get around screening, of things that screening won't pick up. But I think we need to be realistic about the scenarios that we're trying to protect against. We can't protect against everything, but we can do a reasonable job pretty efficiently and pretty cheaply. Operational issues are substantial. In order to kind of make progress on these, we need to do some research. We need to get some test sites out, develop the software, develop the databases. In order to do this, we need to get some funding. I've done this specific work on my own nickel thus far, and I'm pleased to say that I've now got some funding from the Sloan Foundation, and at least 2 of the industry partners, 2 companies in the industry have come together to provide a bit more funding, and they're trying to coordinate a general industry consortium to start looking into this. What's lacking at the moment is government funding, and I've talked to pretty much everyone out there in those agencies. There's a lot of encouragement, but no one's got the dollars allocated to something like this at the moment. So I'll finish there. Thanks.
Good. Thanks, Rob. So we'll next hear from Jonathan Eisen, professor at University of California, Davis, formerly from Tiger. Your heart's still there, right?
Yeah. They only allowed one person with an Eisen name at Tiger, so when Eric came, I had to leave. Although the fact that I recruited you there says something. Right, so while they get this running, I'm an evolutionary biologist, in particular an evolutionary microbiologist, and I'm interested in how new processes, new genes evolve in microorganisms. And mostly I've been working on studies of uncultured organisms and trying to assess them through analysis of little snippets of their DNA, which is how I got interested in some of the issues related to detecting and security for synthetic biology and for related topics. You can see my talk backward. Okay, so this is Drew's title for my talk, but I am talking about some things pretty close to this. So what I want to talk about is a bit taking a step back from what Rob was talking about and taking a little bit of a bigger picture to the issue of security and detection, and trying to integrate what you might want to do at gene synthesis level at the oligo houses with what you might want to do with the rest of the community and the rest of the field. So the main issue is that bad things happen. They either happen on purpose or, as Rob already talked about, they might happen just by accident. And our main goals are to try and detect whether or not this has happened, to prevent it, as Rob was talking about, and to try and attribute it if something does happen. And so I think you can think of this in a lot of different ways. You can take a sort of scenario approach and say, well, we have this good versus bad intent. If that happens, what is the type of organism that might be involved? Is it engineered or natural? And if it's engineered, there's a bunch of different types of engineering. I think it's also useful to take an approach where you sort of say, what is the end product of something happening? And it might be sickness in the population. And before that, there probably was, well, if we're talking about a pathogen, or some toxin spread in the population. And if it was spread, it could have been natural, right? It could just be in a natural outbreak of tularemia or anthrax somewhere. Probably not a natural outbreak of the 1918 flu, but you never know. Or a purposeful release of something. If it's a purposeful or a release of something that's not natural, it could be accidental, as I said before, or purposeful. It could be, if it's purposeful, someone could have taken a naturally occurring organism like anthrax, and released it, or they could take an engineered organism. And for each of these, you can have engineered organisms that are released accidentally or on purpose, and you could have natural organisms that are released accidentally or on purpose. And for the engineered, we, of course, can have multiple types of engineering. And so when you take this perspective, you can now try and design detection, prevention, and attribution methods that have to cover this whole scope of things. So you have an outbreak, and you need to know, is it natural or is it some purposeful attack? And again, when you want to design a way to distinguish what's natural versus purposeful, you have to understand what are the different purposeful things that people could have done. And so here's an example: an accidental release of some synthetic organism. So you're trying to track that this happened and design methods to detect that, and now you have to distinguish it from the other situations that are going on in here. And so, for example, one of the things that we can do as a community is to say, well, if the community gets together to form policies or to form systems, we can reduce the complexity of this problem. So, for example, if you have organisms that are people doing synthetic biology or, in fact, genetic engineering or even working on natural organisms, tag them in some way or create a registry, this could be very useful because it can aid in attribution, as we all, if there was a database of every single strain that everybody worked on related to anthrax and the entire genome sequence of all those strains, we might have had a better way of attributing the anthrax letters. But if every lab that worked on anthrax had inserted their own particular DNA tag into the anthrax genome that they were working on, And if that's what was used in those anthrax letters, we would have a much better way of attributing at least the point source, not necessarily who did it, but the source of that anthrax if it came from a lab. This, of course, can aid in detection, too. So if you have labs working on particular biopathogens, you can build in the tags that you use into a detection scheme so you can more readily detect accidental releases or stolen material. And the reason this reduces the complexity greatly is because it changes this basic decision tree graph where we eliminate this component for at least for the attribution part, and maybe even for the detection part. That component gets a lot easier. So now, if there's a sickness, we might have taken care of how we would determine if it was an accidental release of something in a lab, and we can focus on methods to do the rest of the tasks. So the rest of those tasks come down to a very, very simple thing: distinguishing natural occurrences, natural things that happen in the world, from purposeful attacks. And this is still, of course, not a very simple thing to do. So again, we have a purposeful release of a natural organism, so we need to distinguish this path, if we're at the sickness stage, from naturally occurring things or engineered things. And in addition, we need to think about how we would design prevention, attribution, and detection methods along this path. So let's think about this for a second. How would you detect the purposeful release of a naturally occurring organism? Well, this would mean, if this happened, basically the organism shows up causing a disease somewhere or causing illness somewhere. It's out of place and out of time. It's a natural organism, so you're not going to use a genetic method per se to detect that it's unusual, but it would be in the wrong place at the wrong time. It could be an outbreak. It could be something you're detecting with the BioWatch system, scanning for spores of something. It could be something you detect in the water. It could be something you detect. I mean, it could be anywhere, basically. But so this is pretty difficult to actually figure out how to do this. We need a field guide to microbes, all of them, or at least the major pathogens, with information on their regular patterns in space and time and genetics. We need methods to scan that database and probably some models to understand things that could occur in those organisms that would be unusual. And the example I usually give when I talk about this is the bird watchers that are out there. If a rare bird shows up somewhere, like the American robin that showed up in London a few years ago, in 5 minutes, 1,000 people know about it, and then there's a posting on a new website, American Robin in London, and people know that that's unusual because they have the field guide, they have the list of the classification of organisms, and the American robins don't belong in London. So theoretically, we could do this with microorganisms or select sets of microorganisms, and that, in fact, is what people are talking about doing with some of the really major pathogens, but we probably need to do that with some of the other organisms. So let's take the other possible scenario here. Someone takes an engineered organism and purposefully releases this. This is actually a little bit easier to deal with in terms of designing methods to detect it because we now have the genes that could be unusual as well. So the organism is theoretically different in its genome in some way from anything that could occur naturally. That would be, we assume, the point of the engineering that was done. Of course, we still don't know, we still haven't talked about distinguishing synthetic biology versus normal recombinant DNA methods. So how do we detect this? One way is a sort of a way that Rob gave us an introduction to. We have a database of bad things. So let's say we have the DNA from this organism. It causes a disease. We get the entire genome sequence of the organism. And we'd like to know if there's something weird behind that organism, or it's in a biodetector or something like that. So we can have a database of genes that we consider bad or organisms that we consider bad. I think that this is a useful thing to do, but from a bigger perspective and in the long run, we need to understand what it means to be bad, because then someone could design one of these automated searches of GenBank or other databases to populate the database in an ongoing manner and not have to be constantly curated. It still should be curated, but not constantly curated. Of course, you need a database of what is good in order to do control experiments. So if you want to design a method that can test the number of false positives and negatives, you need to know what are the good things versus what are the bad things. Well, I think we need these things to try and become automated in order to put them out into a field detector somewhere out that's detecting spores that are in the air in the New York subway system, we don't want to have a million people having to check all those results. So the closer we can get to automated, the better. The methods that you need depend on what stage you apply it at. So if you have the sickness happening, you can think of things like looking at symptoms and diseases that might require reporting. Organisms causing illnesses can be tested then for certain things. So you have red flags that you would raise at that stage. You would have methods that you would design at this stage where after the release has occurred, like biowatch detectors, that would be different than those that would be used after the illness stage. Of course, you'd like to have some intelligence behind some of this if you can scan. So that's where community policing might come in. And then Rob's system and other things that can happen at the actual synthesis stage, there are multiple ways you can think of doing this. So Rob talked about one, which is similarity searches for bad things. The Venter Institute, and when I was working at Tiger, we played around with some other methods than BLAST. I think there are probably improvements that could be made on the methods compared to BLAST. But there are a lot of other things you could do. You could look at the bulk orders that are going on rather than individual orders and some general pattern of the bulk orders instead of trying to tag individual oligonucleotides or genes. And of course, community policing comes in here. What I'd like to talk about is, at the end here, is to take yet another step back and say, how do we think of how we're going to do this in a general way rather than a targeted way? Because first of all, people doing bad things might come up with ways to get around our targeted approaches. And if we think of the general approach, it can help us design the targeted approaches too. So we still want to search for bad things that may have happened or, in fact, any sign of engineering. So we have, again, a complete genome sequence of an organism, let's say. And we'd like to search for signatures of some type of engineering. So that seems to be conceivable to me with recombinant DNA methods. You might look for certain restriction site patterns or signals of standard vectors that people might use. It seems to be much harder with synthetic biology unless there is some real enforcement community policing that forces people to use certain types of constructs or alternative genetic codes or something within the community. This might be very hard. I think that the method that has more promise as a general method is to design methods to look for unnatural DNA. And basically what you're going to be looking for is either anomalous mixtures in one organism. So you get an isolate of an organism, and you want to know, is there something that just seems weird within that genome? You, of course, need to distinguish that from natural lateral gene transfer, which occurs in microorganisms in nature. So you have to have some idea what the natural suite of possibilities is. And you might want to do this in communities, too. So if you get a water sample and the DNA from the organisms in that water sample, it might not be unusual to find one type of organism in that water sample, but it might be very unusual to find two organisms together that are not normally found associated with each other. So again, you can think of when you design these general methods for scanning, where you would apply them would give you different methods or different approaches. So at the sickness stage, you probably want to start thinking about unusual symptoms that are not normally found together. That, symptoms of Ebola virus and symptoms of anthrax in the same individuals or multiple individuals might raise a red flag. And again, you still have to distinguish this from natural patterns of variation. The same, you could again go through this whole map here and apply them at the biowatch detector stage. And so how are we going to do this is something that we work on and a bunch of people work on because it's very similar to the issues related to detecting gene transfer in natural populations or studies of the population genetics of microorganisms. It's just turning it over on its head, and rather than trying to figure out what are the natural patterns in an organism, trying to say how can we detect if something is unnatural rather than understanding what are the natural patterns. So you could take a history-based approach. You could take a genome, split it into lots of fragments, try and infer the evolutionary history of each fragment, and look for things that don't follow the same evolutionary path as most of the rest of the genome. And you, of course, need to distinguish this from natural genetic exchange that happens. You could take a structure-based approach. Genomes aren't random in where genes are and where different features are within the genome. And if someone engineers a genome, they might not maintain those natural patterns to the genome, such as locations or GC content variation and patterns like that. I think one of the most promising things is a comparative approach, where you identify the type of organism that you're looking at in some isolate, and then you either have to gather anew, or hopefully you have it gathered beforehand, data on relatives of that organism in nature, the suite of possibilities of the genes and the genetic background of those organisms in natural populations, and then you can build a model of how the natural organism that is related to looks like, and then you can try and say, is there something anomalous about this new isolate? There are other issues that I think are really important to start to think about, including testing methods. So any method that someone designs that's out there, and let's say we come up with some community standard, how are we going to make it a community standard? We need to figure out if they work. So we need positive and negative controls. I think we need a lot of simulations of things we can think of that someone might try and do at each stage, like spoofing the system. And I think we could try something like competitions. You could take real data or simulated data and ask people to try and detect whether or not that's something good or bad within that data set. And again, reminding you that the major goals are to detect things. Well, these are some of the major goals. Attribute them, or if you really can get something to work well, you can possibly prevent them. And I think to do this, we need to really integrate across the whole suite of all the possibilities about where you would detect something happening, about what multiple methods that you could use, and multiple scenarios that could be done, and then try and integrate that all together so that for any possible scenario that you can think of, that somewhere in this path there's at least a detection scheme or an attribution scheme, rather than focusing in on any one of these stages. So that's it.
Okay, thanks, Jonathan. So, Drew, you're going to wrap this up with another 10 minutes. Do you need slides?
No, I'm just going to be real quick. To Steve's points from earlier, and the presentations just from Rob and Jonathan, I'd like to submit for consideration that we try and put in a declaration coming out of here some organization of our support for implementing a screening system of some form that's open and a process by which we can recognize and address some of the issues and concerns around that. And so I'm not going to take a lot of time. I just want to mention some of the additional issues that have come up in our discussions prior to today regarding the implementation of this. So one is a statement to the effect of what doesn't the screening software or platform do? What problems is it not solving? Two, discomfort regarding the idea of putting a piece of software in a position of sole responsibility for inferring the intent of a human. And so how do we best integrate something like this with the successful existing frameworks that have worked over the last 30 years, which we were reminded of last night? Three, barriers to entry. If you're interested in setting up a new DNA synthesis company, would putting in place something like this make it harder to start? Four, barriers to access of the technology. If we put in place a system and only individuals who are at particular institutions would then have access to the DNA synthesizers, would that create pressures that led to the establishment of gray or black markets, pushing people outside of the system? How do we prevent that? Five, process for conflict resolution. If somebody would like to make something, it's being disallowed. They're really adamant that they want to make it. How do we resolve that? How do we improve what we're doing? And six, as was mentioned, the widespread adoption. So a challenge to us is to see whether or not we might be able to come to some consensus declaration around this discussion that we could take forward leaving the meeting.
That's great. So let me see how we're doing for time here. It's approaching 10 o'clock. We could So we're about to assemble up here on stage. We have a panel. There'll be some more remarks, but why don't we take some time now to field questions from the audience directed at our morning talks. Randy, and if you shout, can you shout? And
so I have a few comments just to this. First is we live in a network age, so I think the idea of a piece of software that sits in one place makes decisions not our best technology available.
oh yeah. Oh, Randy, could you just introduce yourself, identify yourself?
I'm Randy Redford. I went to Stanford Biological Parts of MIT, and of course I have a computer background also. So that's the first part. Is I think if we're not using the network, and we're having the idea that there'll be a program at some synthesis company that figures it out, then you're vulnerable to people spreading the problem around to lots and lots of different synthesis places, and increasing the noise level and decreasing the signal. The second is there seems to be a serious issue with the question of whether it's okay to put proprietary sequence synthesis orders out into either a secure or into the open world because of proprietary information. I'd like to point out that as a country, we are now accepting warrantless wiretaps on all international calls, and we are quite willing to track all the internal phone calls, and we're willing to accept other secret systems that we don't know about yet. So and we don't want to know about it. And it would seem like putting the proprietary synthesis orders into the public publishing them might be a fair exchange. If one did that, one could have sequence detection at home, where your personal computers run a distributed system that goes for troubling all the synthesis orders that are all over the world. A second thing is you have to assume that the bad guy is smarter than you. I don't think we tend to do. We tend to think the bad guy knows less than you do. If you assume that he's smarter than you are, and you put out a system which is fixed, version 3, he'll figure out what's wrong and go after weaknesses.
Okay, thanks. We got two.
Question, these are all the scientists. I think a question might be for Robert Jones. You talked about the danger if this is sort of not handled carefully of people bringing their synthesis in-house. I just wondered if you could speak a little bit about the economics of that, because I kind of imagine that actually, most labs are going to do that. Maybe, the Amgens and the Joneses of this world might do that, but actually, economically it'd be a barrier for most people. So first, I'm wondering from a security point of view, is that really a major concern? How many people are actually going to bring it in-house? But then I guess from a viability of the business point of view, if the big companies take it in-house, does the whole DNA synthesis business collapse economically because there aren't enough smaller orders to keep it in one go? So maybe you can address that.
Absolutely, and that ties in with the previous comment. The kind of sensitivity in the industry is at two levels. One is the DNA synthesis vendors directly, and the second is the industry customers who provide the bulk of the orders. Although I agree that having a centralized server to handle all of this could be very efficient and could actually allow you to do some quite sophisticated pattern matching to see if someone is trying to request oligos for smallpox from multiple vendors. Handling that at a central location could be really useful. That demands that the synthesis vendors give up some of their confidentiality. And like I say, that relationship between vendor and customer is really very, very delicate. I think even though it makes sense computationally to centralize things, I don't think that would happen for a moment. So big pharma or biotech companies are submitting lots of orders into the synthesis companies are going to be very sensitive about where that information goes. Like you say, Amgens and Genentech could definitely and may well do handle that stuff in-house. If you were to do anything that took away the bulk of the large volume orders from the synthesis companies, that could really hammer that industry. And like I say, that as much as the industry representatives are driving down the cost, the pressure on prices is resulting in very low or non-existent margins, and so it's a really very delicate industry at the moment economically, and anything that depresses that is going to cause issues. The concern, aside from the economics, is that by setting up screening, we're going to push the bad guys out to doing DNA synthesis in a garage on a second-hand DNA synthesizer, and that's a real concern. But one of the things that we're doing, if that happens, is that we're adding another barrier, perhaps not a very high barrier, but we're adding another barrier to make that work more difficult.
Okay, thank you. We got you here.
So just 3 points.
And you are?
Sorry, Eric Titus, University of Hawaii. Once, I guess, already sort of coming to the fore, that is an idea of rather than ordering these, all of those entirely parts.
You have to speak up. So.
Synthesizing them with the machine at home in the garage, and not with new equipment, but with stuff that will eventually be available, say, on eBay. The second point was the idea of being able to deliberately the screening process, and you touched on that a little bit, but having worked in this area, I can see ways that simple programs like Flask would be able to defeat it that way. Perhaps one idea would be to assemble some sort of red team, like is done with computer security, people whose mission is simply to try to get around the kinds of processes that you would put in place to defeat the bad guys. And the third point is that there's been a lot of discussion of viruses and bacteria, and perhaps we should keep our minds a little bit open about what is possible in terms of the nasties. And one thing I think about is biology. And computer biology is not really well understood. It's a little bit controversial, but we're thinking in the future, something we want to keep in mind.
Right. Okay, we have another comment here from.
I'm J.D. Kidwell with Code Economics, and I think we're being a little naive in a different way. I would bet that sometime in the next 10 years or so, there'll be something attributed to synthetic biology or genetic engineering that is a problem out there in the environment. It may be a deliberate release, it may not be a deliberate release, it may not even be, more importantly, a real release. But the information is being spread throughout the world, and everyone's going to say, aha, it's happening. We have no means in which to look at that. It's going to have a huge impact on our industry. The most likely thing that's going to happen is it's not even real. So how are we going to manage information when someone comes up and there's plenty of junk science out there, right? How are we going to manage it when someone announces that they found a bug that was genetically engineered and people are getting sick, and it's just simply not true, and they have their own political or just bad science agenda for pushing that? And there are plenty of publications that's going to pick it up, even in our own community, but particularly the network. What do we do? Maybe that's a question for Eisen, and we need to put that in Johnson's team.
Okay, let's see. We got Roger, and then I'll get to you, Phil, and then you. So, Roger.
Roger Brent, Washington Sciences Institute. I guess my question is to Rob and Jonathan. My question is to Rob and Jonathan is given before one is can a technical approach work at all? Perl scripts and so on with the bad sequences identified. in a sequence? Or maybe more constructively, what is the kind of ecological monitoring world that this should be integrated within in order to make it sensible? I think this is to you, Jonathan. What is the research program which gives us an idea of the global microbial environment into which this is integrated so it doesn't suck?
Yeah, there isn't such. Well,
No, but what should it be?
Well, I'm actually writing a white paper on what that should be. I think that we need a global microbial field guide. I mean, I think that's where this gets integrated. And lots of people are talking about this. They want to do the background, people call it, for detecting something like the tularemia that was detected in Texas. Is that natural or is that not natural? We need an idea of the background. Of course, the global microbial field guide is kind of untenable. It's an enormous task. Just a bird field guide is hard, right? So, but I think we can. Well, what I think the community can do to contribute is to think about, like the issues raised here, not just terrorist attacks as a concern, but ecological threats and the positive side of things that can come out of this, the agricultural side of things, and then that will potentially get some buy-in from the community, broader community, to participate in gathering the information needed to know whether or not something bad has happened. So that's what you need to have happen.
That's great. Great. Phil, I'm being asked by my handlers to encourage people to use the microphone, so there's one right behind you. Oh, it's not on?
Just a couple of questions about hotline. One is, I think communities are surely getting to the point where we should be specifying what sort of hotline screening is needed, otherwise other people will take the initiative. And the second thing is a different hotline that relates to a question I think was raised earlier on in the session about if you come up with a result and you're not sure whether you want to publish it. Do you wait for the editors to decide, or do you actually have a hotline you can go to? And that was illustrated in the botulinum case with TNAS, where there was a clear lack of clarity what people should be doing. And again, I think this community should be beginning to articulate what they think should be done under those circumstances.
Right. Actually, who is? I can't remember the fellow's name from GeneArt. Are you here? I wonder if you could. You mentioned that there was at least one instance where something came across your radar screen in order. You looked at it, and then you turned to the government in Germany. But you didn't tell us anything about the government level review and what the basis for that was. And maybe you could just say a few words about that now. Yeah. Yeah. But you'll need a mic. I think that mic isn't working. Is that so? Why don't you come up here? So.
I'll use that one. So I'm Hans Bügel from GeneArt. We've been dealing with whom to report to and what to do with monitoring since 1999, since that's when we went into business, and we are a clearly export-driven company since we're from Germany, and the market there is only 15% for us, so the rest is going anywhere. And we have a pretty clear idea, so I wanted to put that into relation on whom to report to if there's something coming up, because we have to, by German law, we have to report every GMO we make. We have to monitor every GMO we make. So that means that we have the screening process in place, and we have what's called the BAFA. That's the long word in German, Bundesamt für Ausfuhrkontrolle. That means that's an agency that's reporting to the Ministry of the Exterior. And this institution is responsible for all the exports out of Germany. And they have a small life science group or natural science group. It's very small, really. So they impossibly can double-check everything we report to them. So what we have established with them is a more or less three-way procedure on how we need to screen things and what we need to report when. One is what's the country where it's supposed to be going to. One is who is the customer, and one is what is the substrate. What David was getting to, that was a phosphotriesterase, which were asked for a known customer in India, but we were declined the order by the BAFA because a phosphotriesterase is a dual-use agent. even though the customer was known and he told us that he wanted to make some sort of pesticide research with that. We were declined that, so he had to go somewhere else, which he probably did. And he probably got it somehow. But we have this BAFA, so this is the institution where we need to report to, and it's reporting to the Ministry of the Exterior.
All of our sequences to Germany?
If you want to order them with us, sure.
Right. Right. Hang on there. Hang on. So that's very helpful. Is that process described publicly? What you've just laid out for us?
I just laid out it.
Would it be possible to get a public version of that, maybe via this organization or elsewhere? I think that might be very useful.
It would be wonderful. We've been trying to do that. As I said, the BAFA is the institution we need to talk to, but they are small. I've been addressed to that with the NSAP before, and we've been trying to set up the BAFA to talk to the National Science Advisory Board already.
Fantastic. Good. Thanks. Hang on a sec. Wait, the energy level in the room is rising, but we had a question over here. So.
Hello, my name is Matt Kyle. I was at iGEM last year. I'll be there this year. I'm going to be working in the Gardner Lab probably. And I think Dr. Chu, you were just sort of saying this German organization may already have some framework, and this isn't a new question. I think chemical producers probably have similar concerns about precursors to dangerous drugs like nerve gas. They probably have some sort of regulatory framework in place that we might be interested in investigating. I'm curious if anyone's doing anything like that. And also, I'd like to suggest that maybe we could compel people adding sequences to GenBank to identify potentially dangerous motifs right when they add that. So that would make an easy framework for searching for bad things. You know, you could go to GenBank, look at all the things that are potentially dangerous, and look and see if it's in a sequence that someone is requesting or that you've isolated from the environment. Thanks.
Okay, Oliver, you had a come on, use the mic.
Okay. I understood. I'm Oliver and I'm also from NEC. I just wanted to ask John and Ron about how open you make the screening system, because and the people in this room know a lot more about the history of cryptography than I do, but in general, implementing your algorithm in secret doesn't seem to work very well. Whereas letting the community have a bash at it does seem to work better, but then, you know, this isn't mathematical algorithm, this is mathematical cryptography. So to what extent do you engage the community in testing? How open do you have to make the system to them? I think the system, the entire system, should be completely open. So that is the database behind it, the data that's used, the training sets, the software, all of it should be completely open, because I think that that's going to be the best way to design a robust system to deal with all sorts of different scenarios. Now, of course, that means that it's open, and you have to deal with the issue of that someone can test their method more easily against it. But if you design a better open system, I think there's no doubt it will be better than a design secret system.
Well, I agree in principle with the open source approach. I'm not totally convinced, given the size of our community and the nature of our community, that a completely open approach is quite the right way. But certainly it should be open to anyone with a legitimate interest in working on that.
Okay, great. Let's continue with the open. Oh, I'm sorry.
I'm sorry. Just a comment. I'm very surprised to not have heard any reference to Project Coast, which was it was like has anyone, like a show of hands, ever heard of this? Project Coast, South Africa, in the late 1980s, where there were a group of scientists trying to develop agents. It was a crazy, insane, ambitious campaign to solve apartheid by obliterating the oppressed population in South Africa. Basically, they were attempting to create agents to target the black population in South Africa. Insane, but the real concern there would be in creating some pathogenic agent that would, I mean, you're going to you know, kill people and harm people. Sorry. Is that me? Okay. Well, I mean, so there's a case, just as a suggestion for trying to address these sorts of issues. It's, from what I understand, has already been attempted. One of the key players in that was a guy by the name of Wouter Basal, and they were tracked down. But I mean, I don't know all the details of it, but there's definitely a case study there, and the approach in actually trying to prevent something like that. So just a comment. Thank you.
Let's now proceed with some remarks from our panel and begin with Michelle. So Steve, we've already heard from you, so I'm tempted to come to you last in the panel. Let's begin with Michelle and Jerry, Harvey, and so on. Again, just please introduce yourself and yeah, well, and you had some. If you have some comments, now is the time to share them.
Good morning, Jerry Epstein at the Center for Strategic and International Studies. We're a nonpartisan independent think tank in Washington, working with some folks at the Venner Institute and with Drew at MIT on the Sloan study that you've heard about. I'll say just a few things about that. My own background is I've been looking at the interface between fundamental research community and the security community in a number of different environments. Thank you all for having me. It's been a fascinating 2 days, but I did have to put the costume back on to get into character for this morning, so forgive me for that. The Sloan study, we're looking not only at the community, but we're bringing in other interests and areas of expertise that are relevant to overall question we're asking is what are governance approaches? And I use governance to be a word that doesn't really have a specific meaning, and we mean by that whatever actions might be done by whoever might be best suited to do them to minimize the risk and promote the benefits. So we've had a group of both technical and policy individuals from a number of different perspectives, series of workshops we're working our way through, ultimately leading to a wider meeting, and I guess Craig Venter's slide said that sometime early fall. And our goal is not to come up with what we think the answer is and launch it to the world, but go through the analyses and develop a number of policy options. We'd like to start off with whatever we can say about what you need to know about the technology, but from that, come up with a different sets of ideas as to if you wanted to do this and you did such and such, what might be the pros, what might be the cons, and what might be the consequences. It's an approach that Bob Friedman, a mentor, and myself inherited from when we were at the Congressional Office of Technology Assessment, where they not only did not require us to have recommendations, they wouldn't allow it. And so we think that would really be the initiation of a wider public dialogue rather than us crawling out from a cave and thinking we've got the answer. You want to go? There's a number of things we could talk about. I thought, and we don't have much time, and mostly what I wanted to do is hear from you, but I could say some things about the relative roles of different parts of the community. The Congressional Office of Technology Assessment did work for U.S. Congress. I had a chance to see how they operate. They've got a big red button and a big green button. And if things are kind of going okay, they kind of lean on the green button. Maybe it's a little green button and a big red button. But when they get concerned that a community or area of activity is happening and they don't think people are taking proper account of issues they should worry about, that's when they slam the red button on. Congress is not a subtle instrument. If you look at the Intelligence Reform Act, that was in many ways also the result of a rather hurried process of what happened after 9/11 and what do we do about our intelligence community. Dive down to Section 6906. And you find out language on variola virus. It is unlawful to knowingly produce, engineer, synthesize, acquire, transfer directly or indirectly, receive, possess, import, export, or use, or possess or threaten to use variola virus. And then they go on to say, variola is defined as any derivative of the variola major virus that contains more than 85% of the gene sequence of variola major or variola minor. This arguably comes pretty close to putting you at risk of $2 million fine and 25 years to life for developing smallpox vaccine. And that was probably not developed with full care and consideration of the community. And just as one other example, after the Eckhart-Wimmer polio synthesis paper was published, Congressional Resolution HRES 514, sponsored by 7 members of the House, expressed serious concern regarding the decision by the AAAS to publish a blueprint that could conceivably enable terrorists to inexpensively create human pathogens for release on the people of the United States. So when things happen in this community and there isn't a lot of communication and the political world sees it, antennas go off, red lights blink, and bad things can happen. So I think this sort of meeting is the best defense against that. The active engagement of the people in the field who really are making these advances in conjunction with the various other sectors of society that, if they're not comfortable this discussion is being held, will be happy to go off and do their own thing. And then very briefly, I'll just wrap up. We're talking about who in government to talk to. Where are the people in which agencies can we have this discussion? There are clearly some efforts being made. We clearly need to do more. The formation of the National Science Advisory Board for Biosecurity brings in all the areas of expertise that have some bearing on the security implications of fundamental biology research. And that board exists. Professor Rubin will tell us more about it, I hope. But that is one institutional effort to try and foster this dialogue, with the one exception that that board was given no authority over classified research in the U.S. government. So whatever questions come up in that realm, that won't handle. Where that discussion will take place is a board that is now being created under the new Office of the Director of National Intelligence. That's what the intelligence community has munged itself and rearranged itself and put a new intelligence czar. But they are very interested in biological science and biotechnologies, and they're in the process of forming an advisory group with members of the community. I hope that there are a number of you here who either have been or will be approached by this board to have this sort of discussion in that world. One inherent problem you're always going to have is somewhere buried in the intelligence community, we need to have classified conversations and classified analyses over what threats to this country are. Somewhere out in the science world, you need to have absolutely open, absolutely transparent processes by which you develop knowledge. And when you've got an open world and a closed world, somewhere there's got to be a fence. And whether you push it here or have sensitive but unclassified discussions in science or bring scientists into Intel, you have to manage that boundary. It's always going to be hard because it's always going to be there. But it does mean we have a national focus from our security point of view. We have an international focus from a scientific point of view. And again, we will always have that mismatch. We're just going to have to manage it somehow or other. But we cannot push the boundary all the way to one end or the other. We're just going to have to deal with it.
Okay,
Thank you.
Thanks very much, Jerry. Harvey, do you want to pick up from there?
Thank you. I'm Harvey Rubin. I'm a professor of medicine and computer science at the University of Pennsylvania, and I met a number of the folks in this room through the DARPA program on biocomputation. And I'm a member of NSABB, which was formed. We had our first meeting about a year ago. I think it was last June. We meet regularly every couple of months down at the NIH, and the NSABB is divided into 5 working groups. The mission of NSAB or NSABB is really to address the issues of dual-use research and specifically not to constrain the advancement of science. The idea really is to identify the criteria of dual-use research and just try and figure out, are there any specific kinds of experiments that require special oversight, special restrictions on communications, and special consideration of the conduct of the research. Just because your research has been deemed by somebody, and we'll get to that, dual use, it doesn't mean it shouldn't be done. It's just the NSABB, which reports to Secretary Leavitt and the National Institute of Health and HHS, is to try and identify those kinds of experiments. So the way the committee has been divided up, the committee, by the way, is made up of mostly academics. There are voting groups and a non-voting group. Most of the voting group are academics, working scientists, work on tuberculosis, and other groups work on viruses, toxins, and so on. There are a couple of lawyers, a couple of people from the policy world as voting members. The non-voting members are representation from every agency and every department in the government. So Department of Justice, Health and Human Services, DHS, so on, USDA. They're all represented, Department of Defense. They're all represented there, and actually the Intel community as well. And we're in conversation with some other agencies to bring them in. There are 5 working groups. One is on codes of conduct, and that really does affect the people in this room. The idea then is, can one sort of create a Hippocratic oath and create, as Phil Sharp said, a responsible community, a community of responsible actions. So, and that would be defined by a code of conduct, and we can go through that in more detail. The second group is made up of the communications subgroup. Communications really revolves around experiments are done, it's getting ready for publication, as you just mentioned. How is that best communicated both to the public and to the journals? Who takes responsibility for the progress of a research program through the writing of the manuscript all the way to publication? So the communication subgroup are looking at that in great detail. The third subgroup is actually a group that's trying to define what is dual use. It's not really clear what dual use research is at this point. You know, people say good science that can have bad consequences, so that's sort of a generic term. But there's a subgroup working on trying to define what dual use is. The fourth subgroup is one on international relations. That's a subgroup that I sit on, recognizing that anything that this community comes up with is clearly international and clearly of global importance. If there are publications that come out with several nationalities represented on the author list, and one country says this is something that we really would rather not have published, another country says, go to it, how do you resolve those issues? So, and also coming up with criteria and codes are clearly, it's clearly a global issue. So I invite you all to, either talk to me or Anselm, who's our staff person, is in the audience. We would really welcome international collaboration. It's very important for us to, as a committee, to engage the international community. And finally, there's a subcommittee specifically on synthetic biology, and that's another subcommittee that I sit on. And the subcommittee on synthetic biology is actually pretty focused on the relationship between science and select agent regulations. And you just heard all the discussion about select agent. There's lots of ways to get around select agents, and a lot of new science that could get around the select agents. So we're talking about primary sequence, but what if somebody comes up with, for example, algorithms to design functional sites and active sites in enzymes, for example? You could do that in any number of ways. There's lots of good science about how unlimited that might be in terms of primary sequence structures. So the synthetic biology group is really looking at, are the laws and regulations that exist today, the select agent rule, are they adequate for regulating and thinking about dual-use research, and in particular synthetic biology? From what I'm hearing, and it's very important, sort of one of the reasons I'm here is to take back as much information as I can from this community back to the NSABB. So I welcome, just communicate with us and let us know what you're thinking. And I'm looking forward to the document that you guys are putting together. Do you think, for example, that the select agent regulation is adequate to think about the biology that we've been discussing in the last 2 days? A lot of this, the NSABB really came out of the report, the so-called Fink report, and the heart of the Fink report were the so-called 7 deadly sins, or the 7 experiments of concern. Do you all know what the experiments of concern are? So you want me to tell you about okay, so I'll just let you know very quickly. It's, so the experiments of concern that would sort of indicate that this is a dual-use experiment, a dual-use research, is first of all, is to render any immunizations ineffective or disrupt immunity. So can you design an agent that would now bypass my local vaccine? Secondly, can you confer into a pathogenic agent or toxin resistant mechanisms? So putting any kind of resistant, even resistance marker in a pathogen could be an experiment of concern. Thirdly is enhance the pathologic consequences of an agent or a toxin. So, the mousepox experiments, for example, turn a pretty bad agent into something really, really lethal. The fourth one is to increase the transmissibility of a pathologic agent. And that, that goes directly to the kinds of things that David Baltimore talked about last night. It's sort of presumed now there's some really good experiments that show at least H5N1 infects cells, tissues deep in the lungs. If you can engineer binding sites for something higher up in the respiratory tract, then you've enhanced the pathogenicity, the transmissibility, not necessarily the pathogenicity, but you've enhanced the transmissibility. So that would be an experiment of concern. The next one is to increase the capability of a pathogen to be disseminated, and that has to do with the stability of organisms in the environment. So I'm really very interested in Dr. Eisen's approach of, what's out there in the environment? How do you increase stability in the environment? The next one of concern is alter the host range or tropism of a pathologic agent. The last one in the Fink list is to enhance the susceptibility of the host population. So when you think about that, if I could design an experiment that would make this population more susceptible to an infectious agent, well, that's really an experiment of concern. But think about all of cancer chemotherapy. In effect, designing good cancer chemotherapy, no matter how good it is, makes you as an individual more susceptible to any infectious agent. So, you know, I actually practice infectious disease. We see people with, a lot of the cases that I see in the hospital are people who are immunocompromised. And they're deliberately immunocompromised, not because they're HIV/AIDS, but because we've just given them a whole series of chemotherapeutic poisons. And they are clearly more susceptible to an agent. If I do work to develop a better cancer chemotherapy, does that make my experiments an experiment of concern? Well, you know, we can talk about that. Obviously, I think not. Finally, the last experiment of concern that doesn't appear in the Fink report that really came out of NSABB is exactly in reference to this community, and that's to generate novel pathogenic agents or toxins or reconstitute an eradicated pathogenic agent. And so Drew gave us the sort of the three cardinal examples of that. One is the H1N1, the 1918 flu, and we can talk about how the committee handled those Science and Nature papers if you're interested in that. Those papers came to the committee before they were published. It doesn't exist basically in nature anymore. Secondly is, as Drew said, Ebola. You have to wait for Ebola to reemerge to start studying it unless you call up your DNA store. And the third one is very highly regulated agents like smallpox. And the 85% rule is clearly an example of where anybody who's clever can get around the select agent regulation for that. So that last experiment of concern really arises out of the potential of this community. And it's very important for the committee and NIH to figure out how to handle that.
Okay, that's great. Harvey, you mentioned you have I didn't catch the name. You have a colleague here who's...
Yes, Anselm. Anselm, are you?
Could you, I wonder if you could
there.
Stand up.
Anselm Stewart, right.
Yeah, okay.
So if you have any communications, you can either email either Anselm or myself.
That's great, thank you. Michelle,
Thank you. I'm Michelle Garfinkel. I'm a policy analyst at the Venter Institute. I still consider myself to be a card-carrying virologist, but for the last 8 or 9 years I've been working in policy research. My interests are in science and research policy and the professional conduct of science. My most relevant project before this on those issues was at AAAS, where I was an author of the first stem cell governance report coming out of AAAS. I currently work with Robert Friedman, who with great regret could not be here today. I am a senior researcher on the Sloan Synthetic Genomics project that Jerry just discussed. I'm sort of the utility person on that. I look at a lot of different issues, but the one that's really sort of closest to my heart that hasn't been discussed here explicitly yet, but has been sort of around the edges of many of the really fantastic talks that I heard today. I'm just so impressed with what's going on, is the issue of biosafety. I have a little bit of a parochial concern about this. My desk is 1 floor up and directly over the benches where much of the synthetic genomics work is happening. I feel perfectly safe. Others in our community may or may not feel safe if they knew what was going on. We want everybody to feel safe. The two big issues are worker safety and protection of the environment. Science is a form of, scientific research is a form of labor, and I, as a policy researcher, as a scientist, as a member of the community, care about the safety of researchers. And I think that this is something where, going back to David Baltimore's talk yesterday, he's absolutely right. There has been no danger that we can detect that has come out of typical recombinant DNA. I think a lot of what we're seeing with synthetic biology will be very much like recombinant DNA. There are two big issues, speed and scale. If we can imagine, originally the idea put into my head by Drew Endy, a synthesis farm that is as big as our current sequencing farms, that is going to be a lot of novel constructs. They may not be a lot of novel organisms, but there is going to be a lot of novelty. We haven't dealt well yet with various kinds of novelty, with chimeras, with multiple, taking hundreds of little bits of DNA from known organisms but putting them together. What my colleague Bob Friedman calls the Ben and Jerry issue, a bunch of benign bits mixed together of ice cream is not, may not taste good, but it's probably not dangerous. Is that true of bits of DNA as well? We're concerned also about accidental release to the environment. I am not, in my research right now, dealing with purposeful release. There are other issues there that are general, again, to recombinant DNA and are covered by many regulations. I'm concerned about what happens, going back to John Eisen's talk, when something gets out accidentally. What does that mean for research? What does it mean for the community? What does it mean for society? I think that there are, again, from the talks, the opportunities for good are just remarkable. The safety risks are not zero. They may be very small. We haven't thought fully through yet what those risks are. We are dealing with this as a section of the Sloan Synthetic Genomics project. So any ideas that you have, I would love to hear from you. One option that I am working on right now, and that I think is worth mentioning here in terms of Drew's comment, do we really want to trust software to pick up intent? I think there are ways to link that in with various biosafety oversight mechanisms, including biosafety officers, IPCs. There are ways to do this. So I think that we're not at a total loss for how to put these together and, again, make the research so that it not only appears safe, but is safe. Thank you.
Great. Thanks, Michelle. Steve, do you have?
Very, very quickly, because I want to hear from the audience. Right. I think I hear in the background a tremor that's really exciting, which is that people are talking about writing something, writing code. And there's this subject, there's a whole class of products that you can't contract for or describe in advance, because if I could write the description of the software I needed, I would have written the program. And I think that it would be really, really interesting to actually try and write down these things, because at a certain point, you can't describe in advance what these are, and the way you learn about, whether you can make a better screening program is to try and do it. And I think, that would be really exciting. I think that's the way you take the research project to the next step.
Okay, thanks. So let's go back to the floor for questions, comments. Tom, you want to come use the mic here?
Yeah, my question is whether there is a danger that synthetic biology just becomes the sort of receptacle for all of our concerns about advances in biotechnology, regardless of whether it has anything to do explicitly with synthetic biology. A, do you think there's a risk? And B, what could the community do to mitigate that risk?
Okay. Somebody want to tackle it?
Well, I'm an old lawyer, and you definitely have this issue, right? It ties into sort of long literary traditions in the society about things we worry about. So you're a natural receptacle in the same way that nuclear things were in the 70s, and we're going to have talks about that later. The question really is, what can you do operationally? And I think what you can do is be very, very thoughtful among yourselves and understand exactly what the risks are among yourselves before you go out and talk to the public, because we have this urge to kind of go out and reassure people. But the one thing that your lawyer would want to know is what exactly is the right answer, because if you have a good right answer about what you think about these things, that's got tremendous honesty behind it, and I got to tell you, it's much easier to do that than to ask your lawyer to please make up a good story that will reassure the public. So thinking carefully is always a good technology.
That's great. Paul, did you have a? Oh, I'm sorry.
No, that's going on. Part of this, I think, has to do with a question that came up yesterday. Is this new? One thing we're looking at in the Sloan study is there's plenty of already unresolved issues with recombinant DNA, and they're still going to be unresolved when we're done looking at synthesis. So we're asking ourselves what's new about synthesis to add on to what's existing. In some ways you can't do that because some of the policy measures that might mitigate risk of synthesis are more generic. But we have wrestled with how do we pose this and what's the novelty, and I think there's a running discussion. In many ways this is new. There are some transformational capabilities, there are new disciplines, there are new ways of thinking, but at the same time we are building on a base of scientific advance in many other areas that is already going on. And so to some extent stressing what's new is important because it's what's real about the subject, but in some sense stressing the continuity is important because we as a society are doing things and have assumed risk all along, and they are still there and we're being on top of that. So Blending and balancing the newness versus the continuity, I think, is part of this answer. Another part is that I don't think we can go very far along having discussions among ourselves and then going to the public. We are an open forum. If the webcast had permitted it, we would be on the web right now. Science writers today are different than they were 40 years ago. They are very good at finding out discussions in communities, and the responsible ones then write about that responsibly, and the less so write about that less so. But we are going to be doing this in real time. When we identify a risk in this field because we need to get our hands about it, then people will know. And we have to realize we can't go off into a cave, come up with the right answer, and then launch it on the world. We're going to be doing this dynamically. And I think part of the way to get that done is, as this is something I'm learning from Drew, do that openly, do that transparently, because then we're all not worrying about what's going on in a back room, which makes the security part of this a difficult problem, because there's going to be a back room. It's going to be a defensive back room, we hope, and there may be some offensive back rooms. So I'm just laying that out as another example of this is going to be tough.
Okay, great. Paul, and then Dr. Ho, can you wait a second? Paul, and then we'll come to you. Yeah. Please. Yeah.
Two quick points. First, on Tom's point, both Lauren and I are going to start our thoughts on that issue, so thank you for raising that. We'll come back to it.
This part and whole issue is one that I think we're going to need to talk about a lot more, and I'm going to say something about that. Two quick points. One, the issue of if you're making particularly an open source database of all known and potential pathogens, talk about dual use. I would like to hear some more people address that. If you think that's the issue, then centralizing it, making it available, you're really distributing it then to the hacker culture that Drew and others and Rob and others are concerned about preventing the emergence of, but which seems like it is going to emerge. So that's one side. The other side is, this is just saying it isn't, there's no absolute security response that's possible there. The other side is, before we embrace the German model too much, let's keep in mind that under the same German model of export controls, Saddam Hussein received nuclear materials and Iran has received nuclear materials. So it may be that there's some virtues in federal regulation of that sort, but if you're dealing with terrorism and with issues that are really not the run-of-the-mill, day-to-day sort of business, I wouldn't look to large bureaucracies to solve your problems.
Okay, I'm sort of we've got this challenge in this kind of discussion of going deep and staying wide, and I'm just tempted to. I want to get some more feedback from the audience now at this stage, because we'll have the rest of the day to keep expanding on various features. So, Dr. Ho, please.
Yes, thank you. Especially, I want to continue from the say whether that is new. I do think there's a new difference between what the recombinant DNA did before and this synthetic biology, because you tackle a new area. Before, whatever recombinant DNA mostly get information what we did not know at the molecular level for different organisms, and also maybe added some gene, something produce some good products, medical, mostly benefit medically, and later on, like my work, maybe more broader products. But now, synthetic biology, we get into a new area. Before, whatever you not modify, you're not changing the physiology of an organism. But now you intentionally wanted to do that. So I just want to, I'm totally for science, for progress, but it's just a little precaution, because we may redirect evolution to too fast, to a wrong direction, or to something we cannot imagine. Okay, just to say, get just a little bit careful for that. Okay, that's all I want to say.
Okay, thank you. Nubar.
So I just want to comment.
And again,
Nubar from Flagship Ventures.
Thank you.
I just want to comment on this discussion that Professor Ho just mentioned, because I actually view this in the exact opposite way. I think what concerns me far more is actually the sequence information and the cost by which we will certainly be able to read sequence information, is going much, much faster than, frankly, synthesis costs and cycle times will. And one of the things David Baltimore said yesterday, which is, you have to make viable organisms. Well, it seems to me that the evolutionary techniques we've been using in the lab for the last 20 years, I heard the comment this morning, normal recombinant DNA versus synthetic biology. I was stunned to hear normal recombinant DNA. These are techniques that have been available to us, that have been perfected, and I would argue that you could get any kind of viable sequence that's available, provided you have rapid sequencing capability, far more easily than putting in 3, 4, 5, 10, 20 new genes. So I'm not saying I'm not worried about that, but I actually think that the capability to do what humans have been doing a long time, which is breeding, but now done at the molecular scale, is here. And the question that was said, are we bringing this on ourselves? I think this is new, and I think that's wonderful. But in reality, this is a problem that transcends synthetic biology. This is a problem that the knowledge we can get is getting so much cheaper that the techniques are already available to make viable organisms without using what we now call ordered DNA from vendors. I think that's the least of our concerns.
Okay, thank you. Michael, do you want to, Mike Stebbins, do you? Sure.
Does it stand in the power? Do we?
No, I think maybe if you, Harvey, if you come over here, and maybe just you guys go ahead and tuck yourselves. Rob, where's Rob?
I'm safe.
I'll get you. Oh, Rob.
If you want me to wait,
yeah,
I can.
While we're setting up, I didn't see you, Rob. Rob Carlson.
I just have a couple of accumulated thoughts, I guess. And sorry, Rob Carlson, University of Washington, and Bioeconomic Research Associates. Many of the things we're discussing here sort of fall into the realm of social engineering rather than physical implementation of security. Or the physical implementation of security is built on the idea that somehow social engineering is going to work. And the last few days have been full of discussions of how hard it is to engineer even a single cell. So I just want to, in that analogy, raise the question about whether we can pull off any kind of social engineering on this very disparate community that might have a hope of being successful. And then in contrast maybe to what Nubar was saying, or at least in addition to what Nubar was saying, the physical security side of things is really, as far as I can tell, still dramatically, what's the word I want? We're not doing a very good job at it, I guess. While it is possible to sequence from a large sample very quickly, if we really want to do environmental sampling, or if we're really worried about organisms that pop up, we have to sequence and understand very small samples and do it very rapidly, and we still don't have those tools. So if you want to build a database that somehow compares sequences that we find in the environment to dangerous things, that's great, but we still don't have the tools to make that happen in real time. And I think that fits into a larger fraction of something that I saw missing throughout the meeting. We keep talking about trying to make biology more like engineering. Well, every mature engineering field has a significant contribution to it, or a significant component to it, that's test and measurement. Right? All of the gadgetry that makes engineering work. And we didn't hear anything about that in the last few days. That's all.
So that sounds like a technology opportunity, among other things. Okay, thanks for that. Michael, go ahead.
So I'm Mike Stebbins. I'm the Director of Biology Policy at the Federation of American Scientists. Most of you probably haven't heard of FAS. Traditionally, we've worked on national security issues relating to nuclear weapons and radiological weapons, but we had a significant working group on biological weapons convention. And so I'm going to talk about two of the projects in my group, and then and then something at the end that will relate directly to what we've been talking about today. But this will give you some background. We have a biosecurity and biodefense resource. This is actually an earlier one. We don't have a wireless here, so but this page basically supplies you with people with information about who's working in biosecurity policy, information about threat agents, international organizations, reports, and legislation are all on here, and there's an interactive biodefense research map with locations of BSL-4 facilities. So this is the basic framework for our web presence. That's significant because FAS's website gets about 2 million hits a month. So we have a significant travel here. one of the main projects that we have is biosecurity education and particularly relating to dual-use research. And what we're doing is building a curricula that can fit into ethics classes for graduate students or however people want to use them, essentially to raise awareness of dual-use research issues within the scientific community. And the charge was defined certainly in the Fink report in 2003. That was one of the needs that they defined. And then NSABB, of course, has been working on this issue as well. So our strategy was this is funded through Carnegie Foundation. This project is multimedia presentations that use real case scenarios, including the poliovirus scenario, and we'll talk about some of those as well, that spark discussions, and they're computer-based right now, and we're also going to be producing print material and PowerPoints so they can fit into anyone's teaching style and be implemented in any way people like. It's for the certainly with the experience range of advanced undergraduates through postdocs. We have the primary research papers in each one of these, the video clips of the scientists discussing the science itself, the implications of it, and some of the aftermath of it. And so you're getting the scientist's point of view on the dual-use research that they were involved in. And animations to explain the experiments. Again, whoever's using it can read the actual research papers, news stories that came out afterwards, reports that have come out that are related to it. And so it basically fits in and is very flexible. So the first 4 modules we did were a poliovirus case study that we've heard a lot about from Eckhard Wimmer, the mousepox virulence study that was mentioned earlier as well from Ron Jackson, inhalation drug study as well. But you'll see most of these are relating in some way to synthetic biology, so particularly DNA synthesis technology. So this is the general design of it. This is the front page of it, where you have introduction and then each one of the case studies, walking all the way through. These are just some screenshots because we don't have the actual internet running right now. But basically, they're fairly easy to walk through. Gives you a history of the research that's going on, a description of the actual experiments, the reaction, discussion, and references. There's no real judgment. It's more like this is just what happened. It's sort of a documentation of what happened relating to some of these dual-use studies. So here's a screenshot of Ron Jackson, who was discussing some of the unexpected results in the mouse-pox experiments. And again, we'll be making print material. And now we have plans, we have a collaboration to do this there'll be a Chinese translation of it, and that'll be moving forward in the next couple months as well. And we'll be filming some new ones coming up as well. We'll be evaluating them, but the next one we're going to film, I think next month, is Jeff Taubenberger will be doing it with us. Maybe something on RNAi and antibiotic resistance. So that's the general framework for that project. But what we also are going to put into these modules is some of the recommendations from the efforts from the Sloan groups, and of course this community as well. So going along with that, there are some technical issues that keep coming up. What to screen against is one of them. So we're actually going to have, and particularly, how large of a window, what should be involved in this sort of screening for ordering of DNA and oligos. And so basically what we're going to do is hold a workshop so that stakeholders in this area, the synthesis companies, people who work on select agents, government, and certainly the computer programmers can come in, and we'll actually try and define what exactly these technical problems are and resolve some of them. At which point we'll come up with, we'll actually release the conclusion of that and put it out for public statement and basically make recommendations off that. So it's to complement the work that the Sloan Group is doing and to try and actually move forward in that area as well. So That's basically what we're involved in now. So this is sort of next steps when people are talking about biosecurity, integrating it actually into curricula and moving forward with some of the policy efforts is incredibly important. So this is sort of the front face of what's going on behind the scenes in a lot of this area. So, and thank you.
Great, thanks, Michael. Thanks. Jim Newcomb from BioEra has some thoughts he wants to share with us.
Yeah, thanks. Eric invited me to say just a few words about our work, which is bringing an economic perspective to synthetic biology. There's been a lot of talk at this conference about orthogonal systems, and the perspective of economics is, in a way, orthogonal systems define interesting spaces, and in this case, bringing the intersection of biology and economics is an important one, especially as one engages these policy debates, governance debates around this issue, which tend to define a quite narrow envelope around risk, as most of this discussion has focused on the fabrication and dissemination of particular risk agents. There's been almost no discussion of the benefit side, for example, of fast response systems, of all the other potential economic benefits, not only in terms of robustness for public health, but other economic benefits that have to be a part of the equation when we look at the balance from a policy and governance perspective as a whole. So our work is aimed in that direction. We're working under contract from U.S. Department of Energy to do exactly this. It's admittedly an early stage in which to try to assess the potential economic benefit of work in this area. One has to really take a fairly general systems perspective to look at that. I just want to let you know that one part of that project has entailed the creation of scenarios as a way of structuring our thinking to kind of open up the envelope of what the major drivers may be that would influence the dimensions of economic and social benefit. And so we'll be coming forward really as a vehicle for discussion with a set of scenarios that we hope to bring out to this community as a way to engage and widen that debate. I haven't talked to Drew or others yet about whether synthetic society or other platforms may be useful ways to engage that conversation, but that is certainly our intention to put forward some template scenarios as a way to begin that discussion, and all this work will ultimately be incorporated into a report that goes forward to DOE and the GTL program later this year. So we'll welcome your contributions and ideas and input, and I know many of you have already participated in some of the interviews for that project, but we'll look forward, and if you have other ideas on ways that might inform and extend our work on the economics and benefits, please come talk to me.
Thanks very much. Dr. Krasnig from the European Commission, I wonder if you wouldn't be willing to just say a few words to introduce people here to the existence of a formal program. It's the only formal program that I'm aware of, that is a government-funded program called synthetic biology, and what activities are underway now in this broad area of safety and security and risk assessment.
Is this mic? Yeah, it's working. We have. I've just introduced myself. My name is Christian Krasnik from European Commission, and I come from the research funding side. So our idea was in this research program, the framework program for research in the European Union, to fund emerging fields. And one of the emerging fields that was were identified was synthetic biology. And I would say that first point is that compared to the size of the community, of course, the US community seems more mature, and we were starting with, would say, an offer for research funding, and compared to the offer, I think the demand was quite low in the beginning. That was 2 or 3 years ago. in the meantime, I think we could establish a kind of small community, and we also fund, aside of research projects, we also fund other activities. One of the activities will be the next year's conference on synthetic biology, which is part of the series of conferences now. This conference will be in Zurich, Switzerland. Another activity is looking at an analysis, research communities between North America, Europe. Another activity, and that's maybe a contribution to this specific session, is we'll look at the risk perception at European level and also dealing with biosafety. I think the coordinator of this study is also available, so if you want to speak to him, this would be also an opportunity for you. In terms of other policy issues at European level, especially biosecurity, I must say that the common security policy I'm not a specialist for that, so I'm just apologize for that, but the common security policy is, would say, is in a quite embryonic state. It's not really established yet. It has started in last years, and it's outside the really integrated pillar of the European Union policy. So it's one of the pillars that is still handled by bilateral or multilateral agreements within the European Union. So I spoke to some people dealing in the European Commission with security issues, and I must say that biosecurity is a topic, but I'm not sure yet whether in terms of synthetic biology there might be really new issues coming up in the European perception. And maybe there's also still a lack of awareness what synthetic biology is, but or what the new developments in terms of DNA synthesis could bring up in the next years. So I think that within the framework that we have now in Europe in terms of biosafety, release of GMOs, most of the issues seem to be covered in the European understanding. So biosecurity, I think, is something, of course it's interesting to hear here and listen to your discussion, but I think biosecurity is not that hot topic at European level. Might be different in some member states, some of the European Union member states, but at European level this is something we still have to look at in the future. Okay, thank you.
Yeah, thanks very much for that. Any more questions, comments from the floor, from the panel?
Just one. I really liked Paul's comment about this open source question, and I think it's an interesting empirical observation that for the last 3 years, hackers have not actually gone out and discovered bugs in code to build viruses around. They reverse engineer the Microsoft patches. So this is actually a subtle and interesting subject that's been well studied, and I urge the people when they design whatever they want to design to actually find out the learning about that, because you can't just do this on sort of ideological grounds. You actually have to think it through, and there is a body of law about, of thinking about quality issues and how you design those things. The other thing that's kind of nice is the one thing you know is you want to have a stochastic program, right, so that you can't challenge it once and know that you can sneak past it afterwards every time. And that's a kind of natural thing to engineer. If each company did a little bit different version of the basic open source process, you could design it so that you just wouldn't know for all time, that it would evolve like these things David Baltimore talked about last night, so that you never know when your next submission is going to be caught or not.
George and then Roger. And then
so, is it, hello? Yeah, so I think the open source issue is, it's a little complicated to have a stochastic version of that, because if they have an open source, they can run it themselves as much as they want, and they can reverse engineer it and so forth. I'm all in favor of open source, most of what we do, but I think in this case there's a need for both an open source version and a closed source version, and that will almost inevitably occur. And that way people can't be absolutely sure they're evading all surveillance and the open source provides a minimum standard above which the closed source has to operate. The other thing I would ask for, a little sort of a similar vein, a little more rigor when we say that we have false positives. When we have, I think that those should, you should say false positive with what screen. Like if you can say false positive before you've done translation and look at good agents, that's different from saying false positive after you've done those things. And I'd like to see a specific example of a false positive when people say that, because I think that if you order up an oligonucleotide that or a DNA sequence that's a false positive after you've gone through all the reasonable screens that a human being would do, I'd really like to see some examples of that, because I haven't actually seen any yet. And also the same thing goes for pharmaceutical companies that would not want to go along with what is beginning to emerge within the industry of the gene synthesis companies of checking in a way that's secure for their proprietary information. If that security can be, is present, then I'd like to see some examples of pharmaceutical companies that have actually said that, rather than just our concern for them.
Okay, thanks, George. Roger.
This relates to George's.
Identify yourself.
Roger Brent, Molecular Sciences Institute. This relates to George's last point. I'd be interested in hearing from the person from Germany, from GeneArt, how the phosphodiesterase, if I got that right, came to their attention. Who flagged it and how? Because that obviously is not a pathogen. Phosphotriesterase, big deal. Okay.
Thank you. I can comment that it was flagged by one of our bioinformatics scientists who actually knew what it was and what it did. So it's not getting back to, do we want the software to detect what's pathogenic or what's bad? No, we don't. We want the software to help us identify, and then we want a scientist and actually people who know the science, who know what's going on, to have the final call on that.
So Roger, we should point out that in fact the select agent rule includes things other than living organisms. It includes toxins. And so, right. So, but it's important to recognize that we're talking a lot about living organisms, but there's another whole side that includes chemicals and toxins and so on. At least half, right.
Okay, so I think Drew has some closing remarks, and then we'll move into the break.
Just want to say I very much welcome any input you have on this, but if you use the email address in the information you have, it won't work, because I don't now, unless Eric wants to hire me, I don't work at Tiger. But gepstein@csis.org, but even safer is to send it to Michelle, because she'll actually keep track of it.
One quick comment, correction of fact, and then I'll try and make a proposal that might close this session. With respect to a hacker community coming from MIT, this is viewed as being good. And I just wanted to state that explicitly in the context of biological technologies, because this will be webcast. Some members of that community are likely to watch this on some time scale. And I'd very much want them to feel like they're included in this part of our discussions in our community. So one of the lessons from the Goldman report, and if you look at where we are now with respect to 2 years ago, where 2 years ago we were charged with wanting to talk about stuff, we're in a position, I'd argue, to take the next step and implement some plans of action for making progress. It's not that we now have obvious answers to all of the issues that were raised. In fact, I think more issues were raised in our conversations than answering any of the ones that we started with. The two specific proposals I'd like to see us put into declaration coming out of this meeting would be, first, to make an investment of our political capital and resources in improvements of a technology platform. We heard of this from Rob Jones. In his presentation, he mentioned that with support of the Sloan Foundation and some members of industry, which to name them are Blue Heron and Codon, we might have an open community-wide consortium that would improve our technologies. And so I'd like to submit that we consider including that in the declaration. A second component to include would be, and this is to the Goldman report's second point, but now in the context of Jonathan Eisen's presentation, that we also focus our attention and resources to improve our framework by which we consider these issues, looking at them from a broader perspective. In this case, the plan of action would include making good use of the resources that we just heard about from FAS and Mike Stebbins, and hopefully also the involvement of Jonathan Eisen. So I'd like to put both of those forward and hear if there are any objections or issues that haven't come up, and to see if we might take that out of this meeting, put it out to the rest of the world, but also come back to it ourselves a year from now in Zurich. It's an excellent point. I think, as was mentioned, one of the first issues around this is misrepresenting what it's actually good for. Having said that, I would very much like to get past a point where I'm spending my weekends trying to explain to reporters why they should not be doing things like trying to synthesize smallpox to show that it's possible to do that. So that's a very trivial justification for doing it, but I think, you know, with the best is the enemy of good as an argument, and with all care paid towards not misrepresenting what we're actually accomplishing, you know, so with all that worked in.
So, are you calling for a declaration that affirms that particular framework or a framework? Because if it's that particular framework, I had it say at least 700 questions about it. But if you're saying we should proceed step by step towards a framework.
Correct. Yeah, let me clarify that, right? So not any one specific thing that was discussed here in terms of technical implementation and whatnot, but rather an operational process with our support behind that, with points of contact, right? Rob Jones, for example, as one person, that we would hold accountable over the immediate future to make some progress, report back, and see how it's going, right? Not that we've figured it out, but rather we have an operational plan for implementing making some progress besides just coming back in a year and having another conversation about what it is that we might actually be doing, right? Let's go get something done. So not we explicitly endorse using BLAST, right, to do this, right? But rather let's pool our resources and try and make some progress. Sorry.
Microphone.
Drew, I was impressed with those 4 things that you had set up a while ago, your second round of speaking, and maybe you could just return us to that so we could focus on those 4 things as possible areas of agreement.
Points of concern with respect to doing this?
No, the things about, you know, agreeing that there would be a mechanism and a process, and agreeing that there would be, we would attest to the fact that there were gaps in the knowledge, and there were 4 considerations that you had raised that we thought were smart. And we hope you remember
This
them.
doesn't solve all problems.
Yes, yes.
It could be perceived or actually be exclusive to others trying to enter the industry. It could provide a barrier to accessing the technology that would create pressures leading to the establishment of gray or black markets that might otherwise not come into existence. As to Roger's point, it could be misrepresented as solving a problem that it actually does not. And so on. I forget, you know, the next 6.
But those were good first 4.
Yeah.
I think we could,
so again, we're not signing off on we've
Right.
got solutions to them, but rather these are the sorts of issues that we're going to hold the folks who we're supporting to make progress on this accountable for, right? And we'll see how it goes.
So nobody's mentioned the measure that I know at least Ray Zelinskis has been urging on everyone, and that is be kind to your graduate students and postdocs. So you'll mention that. So we should wrap up the session. I think it's been a really healthy discussion. Let's thank the morning speakers and the panel. And we can. Right, we'll be continuing this discussion. So we now go into electrolyte balance mode and some break, and we'll reconvene at 11:30. If you're speaking in the next session, please come forward with your to load your slides.